Dear student, I am Dr. Prakash Mungli, Professor of Biochemistry. In this lecture, I will be going over regulation of eukaryotic gene expression. Regulation of gene expression is an essential feature in maintaining the functional integrity of a cell. Increasing or decreasing the expression of gene can occur through various mechanisms, but many of the important ones involve regulating a rate of transcription. So, in addition to the basic transcription proteins, RNA polymerases, transcription factors in eukaryote like activator and repressor proteins help control the rate of this process. These regulatory proteins, they will bind to specific DNA sequences like enhancers or silencer elements which are associated with eukaryotic gene regulation. Other mechanisms are important and gene expression is controlled at multiple levels. Let's look at how exactly the regulation of gene expression will be going on in our cells. So when it comes to regulation of eukaryotic gene expression, so in eukaryotic cells, as you see here in this particular figure, DNA, it is packaged in a chromatin structure, and gene expression typically requires a chromatin remodeling in order to make the desired gene accessible to RNA polymerase and other proteins like transcription factors. These are required for gene expression. So, the important aspect of this chromatin remodeling, this involves transcription factors that bind to the DNA and recruit other co-activators such as like histone acetylase, as it is shown here in the figure. Histone acetylase, it favors gene expression because it causes acetylation of histone, which are rich in positive charges. And histone deacetylases, they will favor inactive chromatin. So, certain lysyl residues in histones are acetylated, thereby decreasing the positive charge and weakening the interaction with the DNA. That's what histone acetylation does. A chromatin remodeling engine that binds to the acetylated lysyl residues and it reconfigures the DNA to expose the promoter region. And additional transcription factors will bind in the promoter region and recruit RNA polymerase. So that's what I have briefly written here. Chromatin remodeling has to be done. So binding of transcription factors and histone acetylases has to occur. Histone acetylation done by histone acetylase, it favors the gene expression, whereas histone deacetylation done by histone deacetylase enzyme, it will favor inactive chromatin, whereby a gene expression is not going on. Acetylation usually happens on the lysyl residues on a histone protein, and chromatin remodeling engine, it is further exposes a promoter region, and additional transcription factors will bind to the promoter region to enhance the transcription or to initiate the transcription process there. Now, once the transcription complex is formed, see the low level of transcription or the basal level of transcription will start to occur, maintaining a moderate but adequate levels of proteins encoded by this gene in the cell. The transcription factors assembled in this complex are referred as general transcription factors. And there are times when expression of the gene should be increased in response to specific signals that such as like hormones, growth factors, intracellular conditions. So in this case, there are DNA sequences which are referred to as like response elements that will bind to specific transcription factors, and several of these response elements may be grouped together to form an enhancer that will allow control of gene expression by multiple signals. And those enhancers are the ones that are mentioned here in this particular figure. So, which are 1000 base pair upstream. It is shown like GRE, CREs, EREs. These are all enhancers which are basically the response elements. And then of course we have promoters like Tata box is there, GC-rich box and the cat box are there at minus 25 or minus 75, that's in cat box. And transcribed region, as you know, it is mentioned with +1 marking there. Now let's move on to see upstream promoter elements. Only the proximity of the upstream promoter element to the minus 25 sequence distinguishes it from an enhancer. Upstream promoter elements includes like cat box, which is around minus 75, that binds to transcription factor like NF1. And then we have a GC rich sequence that binds to general transcription factor SP1. Both of them are above the Tata box, which is at minus 25. Those are referred as upstream promoter element. Coming to enhancers. Enhancers in DNA are binding sites for activator proteins. And enhancers have the following characteristics. They may be up to 1000 base pairs away from the gene, because as you see here in this figure, transcribed region is at +1 and then the enhancers are up to 1000 base pairs away from the gene. They may be located upstream or they may be located downstream or within an intron of the gene they control. So the orientation of the enhancer sequence with respect to the gene is not really that important. The enhancer can appear to act in a tissue-specific manner if the DNA binding proteins that interact with them are present only in certain tissues. Enhancer may be like brought close to the basal promoter region in space by bending of the DNA molecule, which I am going to show later in the slide. And similar sequences that bind repressor proteins in the leukocytes are referred as silencer, just like enhancers. These are the proteins that will silence the gene expression. So there are few examples of the sequences which are known as like silencers here. So characteristics of enhancers, as I said before. So it is like enhancers, they appear to be tissue specific depending on the type of transcription factors they express in the tissue. Enhancers may be brought close to the basal promoter region in the space by bending of the DNA molecule. And similar sequences that bind repressor proteins in eukaryotes are called as silencers. So that is what is shown here in this particular figure. As you see, transcription, and then we have Tata box, GC rich box and cat box. GC rich box and cat box, they may be, and then after that we have a gap there like up 1000 base pairs starting from the transcription. So we have enhancer element binding to specific transcription factors, like we have CRE, GRE or ERE, like cortisol response element, things like that. These are all response elements which are all interacting with the their enhancing factors, thereby increases the transcription process. Coming to transcription factors. The activator proteins that bind response elements are often referred to as a transcription factors. As you have seen here in this previous figure, look at this. So we have CREB, that is 𝐒𝐿 response element binding proteins, then we have cortisol response binding protein and estrogen response binding proteins. So these are all like transcription factors, and typically transcription factors contain at least two recognizable domain, and DNA binding domain and an activation domain. So DNA binding domain binds to a specific nucleotide sequence in the promoter or a response element. Several types of DNA binding domain motifs have been characterized and have been used to define certain families of transcription factors. Some common DNA binding domains include, we have zinc finger domains for like steroid hormone receptors, and then we have leucine zippers for 𝐒𝐿 dependent transcription factors, and then we have helix loop, helix turn, helix, which is like homeodomain proteins encoded by homeotic or homeobox genes. The activation domain allows the transcription factors to bind to other transcription factors and also co-regulators. It will interact with RNA polymerase II to stabilize the formation of the initiation complex and recruit chromatin modifying proteins such as histone acetylase or deacetylase enzymes. So, two types can be distinguished like with the general transcription factors and specific transcription factors. Now, the general transcription factors like in eukaryotes, as it is shown in the figure down here, these general transcription factors must bind to the promoter to allow RNA polymerase II to bind and form the initiation complex at the start site for transcription. And general transcription factors are common to most genes. And the general transcription factor, that is transcription factor 2D with its Tata box binding protein subunit, that is TBP, must bind to the Tata box before RNA polymerase II can bind. So, other examples include like SP1, NF1 that modulate basal transcription of many genes. So, that is what is shown here in the figure, like transcription factor 2D binding with the Tata box and interacting with SP1 and NF1 proteins over there. So, just to recap, general transcription factors, they bind to the promoter. They allow RNA polymerase II to bind and form the initiation complex at the transcription start site. So, these are common to most genes. So, we have transcription factor 2D with TBP, that is 2D binding protein, bind to the Tata box before RNA polymerase II can bind to it. And then SP1 and NF1 transcription factors modulate basal transcription of many genes. Coming to specific transcription factors. So, the specific transcription factors, the DNA binding proteins are given in this particular table here. So, mentioned with the example, we have steroid receptors, 𝐒𝐻 proteins like that 𝗂𝐶 response element binding proteins. Then we have peroxisome proliferator activated receptors, then NF-kappa B and then homeodomain proteins, and their corresponding response elements are mentioned and the functions for each one of them and the protein class is mentioned in the table here. Just to give an overview of this specific transcription factors. So, the specific transcription factors, they bind to enhancer regions or in few cases to silencers and modulate the formation of the initiation complex. Thus, it regulates the rate of initiation of transcription. And each gene contains a variety of enhancers or silencer sequences in its regulatory region. The exact combination of specific transcription factors available and active in a particular cell at a particular time determines which genes will be transcribed at what rates. Because specific transcription factors are proteins and their expression can be cell type specific. Additionally, hormones may regulate the activity of some specific transcription factors. Example includes steroid receptors or 𝐒𝐻 proteins as it is given in the table here. So, peroxisome proliferator activated receptors referred as PPARs are transcription factors that bind to DNA response elements and control multiple aspects of lipid metabolism. Individual members of this family of zinc finger proteins, that is as you see here, in the table, peroxisome proliferator activated receptors, these are response elements for PPREs there and the protein class is zinc finger. So, individual members of this family of zinc finger proteins are activated by variety of natural and xenobiotic ligands. Examples like fatty acids, prostaglandin derivatives, fibrates, thiazolidinediones. So, all these are the ones which are activating zinc finger protein class of PPARs, which are binding to PPREs, that is PPR response elements. Now, the improvement in insulin resistance seen with thiazolidinedione group of drugs is thought to be mediated through their interaction with the PPAR gamma And then clofebrate is a class of drugs used in hyperlipidemia. So they binds to PPAR alpha, affecting different aspects of lipid metabolism. Then thiazolidinediones, which will act through PPAR alphas. So like this, different class of drugs can target different types of transcription factors. Now going into control of gluconeogenesis by response elements. So regulation of gluconeogenesis in response to hypoglycemia and stress explains how these response elements respond to physiological needs of our cells. Like example, glucagon in response to hypoglycemia or cortisol in response to stress. How that is actually regulating gluconeogenesis. Let's look at that using this particular figure here to understand how glucagon and cortisol respond to hypoglycemia or stress in inducing gluconeogenesis process and maintaining the blood glucose level. So gluconeogenesis, as you know, is a hepatic pathway whose major function is to maintain adequate glucose in the blood for tissues like brain and red blood cells, especially during fasting condition. So it also provides glucose during periods of stress. So hormones that activate gluconeogenesis, as it is shown here in the figure, it is glucagon and cortisol. So glucagon secreted in response to hypoglycemia, and it functions via glucagon receptors and increases cyclic AMP. Whereas the cortisol secreted in response to stress, it permissive for glucagon in hypoglycemia and acts through an intracellular receptor rather than membrane bound receptor, and like other steroid receptor, it binds zinc finger DNA binding proteins. Now, how they will work? As it is shown here in the figure, glucagon increasing cyclic AMP, and it's going to increase protein kinase A and thereby activate CREB, that is cyclic AMP response element binding protein, and thereby it will go and bind to CRE, CREB response element. Similarly, cortisol getting into the nucleus and we're going to interact with the glucocorticoid response element. So by doing so, the PPCK, that is phosphoenol pyrite carboxy kinase, this is an enzyme critical for gluconeogenesis process. So that is the enzyme, the gene for that enzyme is transcribed, mRNA is released, and that mRNA undergoes translation to release proteins, that is phosphoenol pyrite carboxy kinase enzyme. So this is how, and then phosphoenol pyrite carboxy kinase enzyme will convert amino acids all the way into glucose, and glucose is released into the circulation. So this is how the response elements will be modulated in response to hormones that are binding onto the cell surface receptors or nuclear receptors. That's how we can use that in understanding control of gluconeogenesis, which is done by response elements. Now, the control of cell differentiation by homeodomain proteins during development in utero can be understood by homeodomain proteins and their interaction and the control of embryonic gene expression. So that is what is explained here. So sequential and coordinated gene expression is necessary for proper tissue and cell differentiation, especially during embryonic life. So the group of regulatory proteins called homeodomain proteins are the major factors in controlling this embryonic gene expression. So each regulatory protein is responsible for activating a different set of genes at the proper time in development. So the regulatory proteins themselves are encoded by genes called homeobox or homeotic genes. So another closely related set of genes is the PAX genes or the paired box genes. So whether it is the HOX genes or the PAX genes, so the mutation in HOX or PAX genes might be expected to produce developmental errors. Klein-Wardenberg syndrome is one such developmental disorder resulting from a mutation in a PAX gene. Now let's look at co-expression of genes. Most eukaryotic cells are diploid, like each chromosome being present in two homologous copies. The alleles of gene on two homologous chromosomes are usually co-expressed. That means both the alleles are expressed. In a person heterozygous for alleles of a particular gene, for example, a carrier of sickle cell traits, two different versions of protein will be present in cells that express the gene. One is, in the person heterozygous for normal and sickle cell alleles, about 50% of the beta globin chains will contain glutamate at 6th position of beta globin, and 50% of them will contain valine at 6th position of beta globin. So that means one of the chain is normal, which is coming from the normal allele, the other is a mutated one. So 50-50 co-expression is there here. And major exception to this rule of codominant expression includes, like on the bar body in women where inactivated X chromosome is there, so that means only one allele is only one copy is active, other copy is not. So when there is a mutation in the other copy, which is like bar body, so nothing happens, right? So in the immunoglobulin heavy light chain loci, like ensuring that one B cell makes only one specific antibodies, or in the T cell receptor loci. So like this, the co-expression of genes, there are certain exceptions other than that. So two homologous chromosomes having two copies, so alleles, both the alleles will be expressed to make a complete quantity, like 100% quantity of protein that is adequate to maintain the cellular needs. So the control of eukaryotic gene expression and protein levels are control point and examples are given here. So inactivation of specific chromosomes or chromosomal region during development, it's about bar body and other things explained here. And then local chromatin modifying activities, gene amplification, specific transcription factors, processing of mRNA, rate of translation, protein modification and protein degradation rates, all that is briefly explained in the table. Coming to gene imprinting in Prader-Willi syndrome. Just to go a little more into Prader-Willi syndrome here. So the genetic imprinting of a few regions results in monoallelic expression. In some cases, this imprinting is according to the parent of origin. The gene involved in Prader-Willi syndrome is on chromosome number 15 and is imprinted so that it is normally expressed only from the paternal, not the maternal chromosome. If one inherits a paternal chromosome in which this region has been deleted, Prader-Willi syndrome results. It can also result from uniparental or like maternal disomy of chromosome number 15. So symptoms of Prader-Willi includes childhood obesity and hyperphagia, like excessive eating, hypogonadotropic hypogonadism, small hands and feet, mental retardation and also hypotonia. So that's all about regulation of eukaryotic gene expression. Thank you.